In this lecture, we consider DNA replication, the copying of a parental copy of DNA to produce two copies of progeny DNA. The basic themes in DNA replication are the two strands of the double helix have a great affinity for one another. The strands are intertwined and in addition are held together by hydrogen bonding between base pairs. The double helix is also stabilized by aromatic stacking of successive bases. During DNA replication, the two strands must be unwound and the strands completely separated. Unwinding of the DNA strands is accomplished by energy-dependent enzyme systems. Replication is highly accurate. In fact, it's about 1,000,000 times more accurate than would be predicted by the chemical dynamics of DNA synthesis. There are multiple proofreading and editing mechanisms that increase the accuracy of DNA synthesis. DNA replication must be very accurate because unlike transcription and translation, it constitutes the genetic heritage of future organisms. Other observations about DNA replication include DNA replication is very rapid. One DNA strand is synthesized in a continuous fashion, whereas the opposite strand is synthesized in fragments in a discontinuous fashion. The net flow of synthesis of the leading strand is 5' to 3', whereas the net flow of synthesis of the lagging strand is 3' to 5'. Even though the net flow of lagging strand synthesis is 3' to 5', synthesis actually occurs with small segments of DNA in the 5' to 3' direction, and then those small segments are linked together. Specialized enzyme systems are used to synthesize structures called telomeres at the end of the chromosome. This slide should remind you that the central dogma of molecular biology concerns the flow of information from DNA, the hereditary storage material of genetic information, to RNA, and then from RNA to protein. This means that the arrows in this scheme do not signify the chemical conversion of one compound into another. In the example of replication, the DNA is not converted into new DNA. Rather, the parental DNA strands serve as templates for the synthesis of new DNA. The DNA double helix is a tightly packed structure in which the base pairs are relatively inaccessible to enzyme systems that catalyze replication. The space-filling model in the center shows just how tightly the DNA is packed. The basic polymerization of DNA requires the following factors: a DNA template that's copied and directs the synthesis of the complementary DNA sequence, the primer, which is the initial sequence of DNA at the 5' end of the new DNA. The primer provides a 3' hydroxyl group which attacks the incoming nucleotide. Take note that RNA synthesis does not require such a primer. 4 deoxynucleotide triphosphates which provide the nucleoside monophosphate unit that's added to the growing chain of DNA. A DNA polymerase enzyme, which is a complex of enzymes that catalyze the process of DNA elongation. Metal ions such as magnesium and several other protein factors are also required. In addition to the core requirements of DNA replication, other factors are required to open up the parental double helix and to accommodate the special needs of lagging strand replication. Those are single-strand binding proteins help keep the strands apart after they're separated. A helicase enzyme catalyzes energy-dependent strand separation. The topoisomerases collectively relax the supercoiled DNA that's created by the action of the helicase. The primase creates an RNA primer region in lagging strand fragments. This provides the starting material to which DNA polymerase III can add segments of DNA. The nucleotide triphosphates provide energy for the action of helicase and topoisomerase. The DNA polymerase I removes the RNA primer nucleotides from the lagging strand segments and replaces them with the appropriate deoxynucleotides. The ligase closes the gaps between segments of lagging strand DNA. DNA synthesis occurs on a template strand of DNA. The new DNA is added to the 3' end of a primer strand of DNA, so the direction of synthesis of the new DNA is always from the 5' end to the 3' end. The primer itself and the newly synthesized material both run in an anti-parallel direction to the template strand. The new DNA sequence is complementary to the template. So, for example, if the template strand has a sequence ATC running 3' to 5', the new complementary strand will have the sequence TAG running 5' to 3'. The base pairing of the template and copy is A to T and G to C. The first slide shows a primer sequence CAC with a free 3' hydroxyl group. There's an empty binding site for DTTP across from the adenosine A residue on the template. Next, a DTTP will diffuse in to occupy the open site, and it'll hydrogen bond with a complementary A residue on the template, resulting in TA base pairing. The 3' hydroxyl group of the primer will then launch a nucleophilic attack on the phosphorus atom of the alpha phosphate of the DTTP. That nucleophilic attack of the 3' hydroxyl group of the primer forms a phosphodiester linkage with the alpha phosphate residue of the DTTP and releases inorganic pyrophosphate. The hydrolysis of the inorganic pyrophosphate to 2 molecules of inorganic phosphate pulls the reaction in the direction of DNA synthesis. The DNA primer is now 1 nucleotide longer, and everything is ready for another round of DNA synthesis. The primer now has the sequence CACT with a free 3' hydroxyl group on the T residue. There's an empty binding site for DATP across from a thymidine residue on the template. Next, a DATP will diffuse in to occupy the open site. The A of the DATP will hydrogen bond with a complementary T residue on the template, resulting in AT base pairing. The 3' hydroxyl group of the primer will then carry out a nucleophilic attack on the phosphorus atom of the alpha phosphate of the DATP. That nucleophilic attack of the 3' hydroxyl group of the primer forms a phosphodiester linkage with the alpha phosphate residue of the DATP and releases inorganic pyrophosphate. The DNA primer is now another nucleotide longer, and everything's ready for another round of DNA synthesis. The primer now has the sequence CACTA with a free 3' hydroxyl group on the A residue. There's an empty binding site for DGTP across from a cytidine residue on the template. This process of DNA elongation will continue with the same pattern repeating itself as long as there are open positions on the template and all of the other reaction components are present in adequate amounts. One key step to DNA replication is strand separation. The tightly wound parental strands of duplex DNA must be separated so that primer strands can bind and the DNA polymerase reaction can occur. We know that the parental strands of duplex DNA must be separated so that primer strands can bind and the DNA polymerase reaction can occur. Spontaneous strand separation is way too slow to permit the efficient replication of DNA. Rapid strand separation is achieved by an ATP-dependent helicase enzyme. The separation of DNA strands is promoted by an ATP-dependent helicase enzyme. The helicase enzyme slides along single-stranded DNA, gradually unwinding it. The A1 and B1 domains of the helicase have a cleft between them that closes when ATP is bound, causing domain 1 to slide along the DNA. When ATP is hydrolyzed, the cleft between the two domains opens up, pulling the DNA from domain B1 toward domain A1. A consequence of strand separation is that additional strain is introduced into the DNA molecule, causing it to be overwound in surrounding regions. The supercoiled regions of DNA that are created by helicase action are referred to as topoisomers. If nothing were done to relieve the supercoiling of adjacent DNA, the action of helicase would be inhibited and strand separation would cease. Topoisomerases function to first increase and then eventually relieve the strain of supercoiled DNA. Topoisomerase II, also called DNA gyrase, catalyzes an ATP-driven formation of negative supercoils, making the DNA more susceptible to unwinding by helicase. Topoisomerase II cleaves one DNA strand of supercoiled DNA, rotates in a controlled fashion around the other strand, and then religates the cleaved strand. The action of topoisomerase I results in the partial or complete relaxation of supercoiled DNA. The DNA sample on the left is supercoiled. It looks like an overwound rubber band. The DNA sample on the right is relaxed DNA. It seems to be very content. The topoisomerase I enzyme cleaves one strand of supercoiled DNA. It then rotates 360 degrees around the intact strand of DNA. It then religates the cleaved DNA strand. This process occurs repeatedly, resulting in the relaxation of the supercoiled DNA. This slide reminds us that DNA replication is semi-conservative. That is, during replication, the parental strands are separated, and each parental strand pairs with one newly synthesized strand. The two strands of the DNA duplex run in opposite directions, 5' to 3' and 3' to 5'. According to the semi-conservative model, both strands are copied during replication, and one new copy goes to each of the new duplexes. The new strands are antiparallel and complementary to the parental DNA template strands. It's been observed that DNA replication proceeds from a set point in that both strands are copied simultaneously. However, enzymatic studies have revealed that the DNA polymerase reaction only runs 5' to 3'. Because DNA synthesis is initiated at a single point, one strand could be synthesized in a continuous manner from 5' to 3', but it was initially unclear how the other strand could be synthesized in the net 3' to 5' direction. Further studies of DNA replication have revealed that the two strands are replicated differently. It's been confirmed that the DNA polymerase can add on to a 3' end of a nucleotide strand, synthesizing the new DNA from the 5' to the 3' end. The leading strand of DNA is synthesized in the 5' to 3' direction, which is the same direction as the mechanism of DNA polymerase. The lagging strand is synthesized by constructing a series of short segments in the 5' to 3' direction and then joining those segments together. Through the use of this strategy, the net flow of the synthesis of the lagging strand is in the 3' to 5' direction. DNA replication proceeds in the unwound area on both strands simultaneously, and the net flow of DNA synthesis is in the same direction. The leading strand is replicated with a DNA copy being produced in the 5' to 3' direction in a continuous manner. The lagging strand is also replicated 5' to 3', but in short pieces called Okazaki fragments that are later ligated to produce an unbroken product. Although the short segments are synthesized from 5' to 3', the net flow of lagging strand synthesis is 3' to 5'. The discontinuous replication of lagging strand DNA is primed by short stretches of RNA, which are synthesized by a primase enzyme. Primer RNA is later removed by DNA polymerase I, which also fills in the gap with the appropriate deoxynucleotides. DNA polymerase III is the enzyme responsible for the replication of both strands of DNA. The polymerase III is a dimer that contains two equivalent regions of catalytic activity. One of the dimeric sections of polymerase III travels down the leading parental strand of DNA from 3' to 5' and synthesizes the complementary daughter strand continuously in the 5' to 3' direction. The other dimeric section of polymerase III travels down short sections of the lagging strand of parental DNA, which have looped around so that they are also read 3' to 5'. The result is that the daughter DNA segments that are produced from the lagging parental strand are also synthesized in the 5' to 3' direction. The dimeric DNA polymerase III can replicate both strands simultaneously because the lagging DNA strand loops around and presents itself to the polymerase in the same orientation as the leading strand. That is, the polymerase reads the parental DNA in the 3' to 5' direction and creates the new strand of DNA in the 5' to 3' direction. The last activity needed to join the Okazaki fragments is a DNA ligase. The ligase uses ATP to close the single-strand nick between the DNA segments. In the first step, a 5' phosphate from one side of the nick displaces a pyrophosphate group from the ATP, producing an ADP residue that's covalently bound to the 5' carbon. As we've now seen too many times, the hydrolysis of the pyrophosphate seems to drive this overall reaction. The ribose residue on the 5' side of the nick is now linked to an ADP residue. The 3' hydroxyl from the other side of the nick now attacks the inner phosphate of the ADP residue. The attack of the 3' hydroxyl releases AMP and covalently links the two Okazaki fragments together by a 3' to 5' phosphodiester bond. The net result of the ligase reaction is to convert the two Okazaki fragments into a normal continuous sequence of DNA. At this point, we can review the overall process of DNA replication. The site at which the two parental DNA strands are separated is called the replication fork. At that point, a number of enzymes interact with the DNA to achieve unwinding and strand separation. The separation of DNA strands is promoted by an ATP-dependent helicase enzyme. The helicase enzyme slides along one strand of the double-stranded DNA, gradually forcing the two strands apart. The A1 and B1 domains of the helicase have a cleft between them that closes when ATP is bound, causing domain A1 to slide along the DNA. When ATP is hydrolyzed, the cleft between the two domains opens and pulls the DNA from domain B1 toward domain A1, separating the two strands. One consequence of strand separation by the helicase is that additional strain is introduced into the region of the unopened DNA in front of the helicase. The added strain causes the DNA to be overwound or supercoiled in that region. Topoisomerase 1 and 2 function to relieve the strain of the supercoiled DNA. The topoisomerases precede the helicase down the DNA. They catalyze the formation of negative supercoils, that is, they relax positive supercoils, making the DNA more susceptible to further unwinding by helicase. Topoisomerase 1 makes cuts in DNA, unwinds it to relieve the tension, reducing the extent of supercoiling, and then religates the relaxed material. There's another protein designated SSB in the diagram, which is involved in maintaining the separation of parental strands. The letters SSB stand for single-strand binding protein. The function of this protein is to stick to the separated strands of parental DNA to keep them from rehybridizing before they can be replicated by the polymerase III. The polymerase III enzyme replicates both strands simultaneously, synthesizing DNA in the 5' to 3' direction. It can do that because the lagging strand of DNA loops around and presents itself in the same orientation as the leading strand. The lagging strand synthesis is primed with RNA prior to the action of polymerase III. After polymerase III has added the DNA to the Okazaki fragments, DNA polymerase I removes the RNA segments and adds deoxynucleotides. Finally, the Okazaki fragments are joined together to form an unbroken strand of DNA. Those final nicks are closed by a DNA ligase. The initiation of DNA replication has the following features. Each strand must be copied exactly once. The timing and location of replication initiation are carefully controlled. Replication begins at a special sequence called the origin of replication. Prokaryotic chromosomes have a single origin of replication. Eukaryotic chromosomes have multiple origins of replication. From the origin, replication proceeds bidirectionally, which yields two sites where DNA is synthesized, called replication forks. The E. coli origin of replication has a length of 245 base pairs. It contains a tandem array of 3 nearly identical 13 base sequences at one end and 4 scattered binding sites for the DNA A protein. With smaller circular DNA molecules, such as the genome of E. coli, DNA replication is initiated at a single origin of replication. Synthesis of DNA occurs in both directions from the origin of replication using 2 replication forks until the entire genome has been replicated. The replication of larger linear DNA molecules, such as human chromosomes, is initiated at multiple sites on the parental DNA. The synthesis of DNA proceeds in both directions, again with 2 replication forks, until the new duplex strands meet up and the 2 progeny DNA molecules are separated from each other. The Science Teaching and Student Services Building is a recent addition to the East Bank campus. The architecture and the integrated artwork are visually stunning. Here's a feature from the outside of the building. Here I am photographing my reflection in one of the surfaces of that same building. Notice how gracefully I hold the camera. The Science Teaching and Student Services Building has this 4-story spiraling sculpture and a central atrium. You could photograph this feature all day and never take the same picture twice. At the bottom of the spiral, there's this bluish-greenish reflective disc that appears differently from every angle. Here's one look at the disc. Around the spiral is this amazing staircase that itself is a work of art. Again, you could take hundreds of different interesting photographs of this staircase. Or maybe you think none of them are interesting. Here we can see how the spiral and the staircase are integrated. In this case, the natural beauty of the architecture is enhanced with a little Photoshop to give it that purple color. I've done this in a number of other colors as well, and they all look kooky. There are these claw-like components of that reflective disc that remind me of Edward Scissorhands' hands. Here's another architectural feature of the Science Teaching and Student Services Building. Are we all prisoners in our own time and space? At least this prison has a great view. Here's a freight barge delivering its load on the Mississippi River. The first snow of autumn gives this photo of the Weissman an impressionistic look. Here's a look at the Weissman from ground level on the Mississippi River bank. During the remodeling of the Weissman, this crane dominated the campus. It must have been 10 or 15 stories tall. How often do you see ice on the Mississippi and green grass on the riverbank at the same time? What's going on here? That combination of ice and green grass was so unusual that I show it again. During my 40 years in Minnesota, I've never seen this before. This row of bike stations looks like a